Hey guys, my name is Noah for those of you who don't know me, and welcome back to another one of my late night last minute adventuresome videos. In this one we're going to be covering the structure of bacteria, the structure of viruses, the peptidoglycan layer. That's too much for one video, but I'm going to do it anyway because I gotta cram it in. It's 10:18 at night and I gotta get to bed soon. I'm going on a trip tomorrow. So let's get it done. What are bacteria? Bacteria are microscopic, unicellular, prokaryotic organisms. Microscopic. Alright, they're microscopic. Microscopic. What does that mean? It means they're really, really, really small. We don't see them floating down the street. That would be weird, it would be cool, but it would probably be bad. So they're really, really small. We can't see them individually. They're unicellular. What does that mean to be unicellular? Well, it means, quite frankly, that they're one cell. There aren't cells that make up bacteria. They are themselves. It's like the founding of this nation. We hold these truths to be self-evident. Bacteria are unicellular. They are self-evident. Alright, they're made up of a bunch of molecules, but there aren't cells inside the cell. Okay? Unless you're dealing with viruses and they're technically not cells. So anyway, I don't want to confuse you. They're also prokaryotic, right? So prokaryotic. What does that mean? It means their DNA is not enclosed in a membrane. If you remember from the last video, you have the cell, you have the DNA. In a prokaryotic cell, it's free floating. In a eukaryotic cell, it's enclosed. But this is a prokaryotic cell, so it's not enclosed. It's free floating like that. Alright, let's get into the structure of a bacterium. We're going to go ahead and look at a rare Bacillus bacteria, I think. That's what it looks like to me. Now flagella are on the back here, and kind of go out like this, and they're it's like a rudder, you know? They kind of move them around, and it's like a rudder for the bacteria. And on the exterior, occasionally you have pili, which are basically for sexual reproduction and the transfer of DNA in a certain type of bacterial reproduction, and I believe it's conjugation is what it uses these little pili for. Now, they're going to be little, basically whiskers on the DNA. Now, pili are normally rare because it's a pretty rare occurrence for that type of sexual reproduction. But anyway, that's not important. Inside of this bacteria, we're going to have some free-floating DNA, like this. You're going to have a plasma membrane and a cell wall and a capsule, which is basically just like another membrane, I think. And then you're going to have some ribosomes. And remember, ribosomes are used in protein synthesis. It's rRNA, and it's basically, you have the ribosome, and if you remember, the messenger RNA is going to feed, or the transfer RNA is going to come over, and it's going to attach onto some of the messenger RNA, and basically the nucleic acids are going to bond with these peptide bonds, and you're going to make proteins. But anyway, they have ribosomes. So basically what that means is you're going to have proteins being made, and it's going to undergo some reproduction, sometimes a sexual. I screwed that word up. Anyway, moving on. Let's take a look. Wow, asexual reproduction. I don't even know where it's 10:22 at night, guys. Just give me some slack here. All right. So if you take a look, you're going to have gram-positive and gram-negative bacteria. We did an entire lab on this. Basically, it's a staining mechanism to see how many layers of peptidoglycan there is. So let's take apart this word. We have peptidoglycan. All right, pepti. What do you see there? Pepti, peptide, peptide bonds. Peptide bonds are between nucleic acids, and nucleic acids make up proteins, right? So this is going to be made up of proteins. And doglycan, glycan. What do we know? Glycan, glucose, sugars, carbohydrates. So sugars or carbohydrates, carbohydrates, right? So it's going to be made up of proteins and sugars. Okay, so here's what's going on. I'm getting near the end of the page here, but you know, getting near the end of the night. So we have we're going to have basically a cell wall on the outside, right? So this is a cell wall. And then we're going to, on the inside, we're going to have a cell membrane. And in between, on a gram-negative bacteria, we're going to have one layer. I'll use it in the same color, I think. We're going to have one layer of this peptidoglycan. And why it's gram-negative is because you're going to stain it, right, with that crystal violet. And it's going to get stained. The crystal violet is going to get into this peptidoglycan. But when you decolorize it, that purple is going to go away. And then you're going to stain it with, I believe it was safranin. And then it's going to get stained pink. And that's why it comes up pink. Now, gram-positive bacteria, instead of having this one layer, you're going to have the one, but you're going to have 5 times as much. So you're going to have 2, 3, 4, 5. All right, you're going to have 5 times as much in a gram-positive bacteria, right? So there's going to be 5 layers basically. And what happens is the purple from the crystal violet, I was using the wrong color before, but the purple from the crystal violet gets in there and it doesn't get decolorized because it gets held in there, you know, maybe 1 or 2 layers worth gets washed out, but it's going to get held in by the peptidoglycan. Now, really quickly, I'm going to try and make more videos about this, but viruses basically have certain different types, okay? We're going to have DNA, right? We start with DNA. It's going to be encased in a protein coat called a capsid, okay? A capsid, sorry. Now, we're also going to have receptors, right? So basically, I drew this incorrectly. So it's going to be in there, it's going to be DNA or RNA, and it's going to have a coat around it called a capsid, and then it's also going to have, I believe it's called the receptors, but I'm trying to figure out what type of receptors they are. Glycoprotein receptors, right? So we're going to have these little things on the outside, and what that does is it helps the virus find the right bacteria to infect. So it's going to find that glycoprotein, it's going to attach on, and then you're going to have this virus going on, right? Another thing to remember is retroviruses. Instead of going from DNA to messenger RNA to DNA, it's going to start with, so this is a retro, I don't think there's an H. It's a retrovirus, right? Retro means basically like old, like a rebirth almost. So it's going to go from RNA down to DNA, back to messenger RNA, and then over to proteins, right? So that's what a retrovirus does. I hope this helped, guys. I'm sorry this was so fast. I'll try and make another video in the morning about viruses and its reproductive state, but we'll see. I'm definitely going to try. Maybe I'll make it. I might not get it uploaded until later, but I hope this helped. Cut me some slack. It's really late, and hopefully you guys do well on this test. I won't be there on Monday to take it. I'm not quite sure. I'm going to be gone on a trip, so I hope it goes well for you guys, and have a good time.